Log on, tune in, find out. Another good idea from Cambridge.
Is it on? Well, thanks for the invite. I'm going to talk about some probabilistic personalization, and specifically I'm going to demonstrate these models or these methods on an application which is cardiac electrophysiology. It's going to be a little bit different than the talks we had up to now because it's going to be about physiological modeling. So before going into the details of what the method is, let me give a small, a brief overview of what the physiological models are. So the three different components are the models, the personalizations, or the inverse problems, or the parameter estimation, however you want to call it. And the last part is the simulations for treatments. So, the idea is in anatomical modeling, it's rather intuitive because we're talking about the geometrical understanding of the images we see. So we talk, for example, about the parameterizations of surfaces, volumes, or fiber bundles. Then the personalization becomes, okay, how do I stop this thing? The personalizations are then segmentations or registrations or tractography, as we've seen before. In the physiological modeling, we're rather interested in the functional understanding. So how does a heart beat? How does electricity conduct through the cardiac muscle? And or how does pathology evolve within the brain? How does things work? How does blood flow within the vessels? So our parameterizations are rather different to our mathematical models or computer simulations, and the personalizations are based on inverse problems, so parameter estimations or model fitting. As I said, the parameterizations are mathematical models, and most often they are given by partial differential equations. For example, what I show here is a descriptive model for the electrical conduction within the cardiac muscle. I will give details more about this model. But the idea is having various observations from the patient or clinically available observations. The idea would be to have individual specific parameters so that we adapt the generic model to a specific case. Therefore, it gives us the ability to create, for example, individual specific simulations. So as I said, there are two parts in this, the patient-specific parameters and then the patient-specific geometry, which I will not talk about because we've already discussed this up to now. Okay. Now, once we create these individual simulations, what do we do with them? Well, basically, the idea is to create some patient-specific treatment or simulations of these treatments. For example, we're talking about simulating invasive procedures, trying to figure out how to best treat this patient, where to ablate in the cardiac muscle so that the problems, arrhythmia will be gone, but we'll ablate or burn the smallest amount of possible region, or some type of predictive modeling, so say if we have a brain tumor, up to what extent has the tumor invaded the brain so we can guide radiotherapy. Now, so as I said, this is rather the global view of where we are, and I hope I gave a small intuition of what things. So I will specifically focus on the personalization. This is going too fast. I should know how to solve this, right? I work for Microsoft. Well, okay, I think it's fair. So, I will discuss a specific problem and show the parameter estimation methodology. So, as the parameter estimation inverse problem, I have to give the model and then the data and then the method itself. So I will talk about the model, which will be of cardiac electrophysiology. So I will talk about the spatial model and its parameters specifically that we're interested in. Then I will give details about what the clinically available data are, the cardiac mappings, the dynamic MR images, and I will focus on how the intuition or the characteristic of this data, such as sparsity and the noise. Based on these observations, I will detail the method, the probabilistic formulation, and specifically focus on the Bayesian inference, and how this can be done in a feasible time without spending 20 to 25 days of computer time. So, cardiac electrophysiology is basically study of electrical activation within the cardiac tissue. So at every heartbeat, there's a pacemaker which creates an electric wave. So this would be, if it's working, right here, which creates an electric wave that passes through the endocardium, disperses within the myocardial tissue. And as the electricity passes through the cardiac muscle, muscle contracts, therefore you have a beating heart. Now, if I were to probe at any location within the cardiac muscle, I would see such electrical signals. Basically, it's a wave that's traversing within the muscle. So the model that we use uses this principle, and it's based on the Eikonal-Diffusion equation. So the model is a traveling time formulation, and we're describing the depolarization times of the electrical activity within the cardiac muscle. So it looks something like this. Starting from a single region where it's given by a boundary condition, we simply solve an Eikonal-Diffusion model. So we have the convection, which is given distorted by the tensors or the fibers of the cardiac tissue, and we have a diffusive nature. Now, why this model? These models are fast, therefore they are clinically relevant, meaning that we can use them with patient data, we can simulate things very fast, and we can create interesting tools for the doctors. If we look at the different parameters that we'll be interested in, one of the main parameters will be the conductivity, given here, which basically tells us how fast the electricity is moving along at what region in the heart. Now, this information is useful because this is basically the tool that gives us the ability to simulate patient-specific activations. And the second part will be where is the actual onset location within the left ventricle, for example. So detailing a bit more on these parameters, because these will be the parameters that I will solve with some clinical data. As I said, the functional conductivity, which is a spatially varying function within the endocardium. Now, why is it spatially varying? Because there's some physical constraints saying that within the endocardium there is a fast conduction network. So when I leave an electric wave in there, it will sweep through the endocardium and then disperse through the myocardium. So this is shown by a high conductivity within the endocardium and a global conductivity within the healthy muscle. Furthermore, we will deal with patient data. So we will have scar tissue or the periscar regions. These will be extra parameters that we'll have to solve for. So ideally, we would like to solve for the whole functional conductivity, but this is rather impossible or rather not feasible for the computational models. So we use a simple radial basis function approximation. So mapping this infinite dimensional problem onto a n+1 dimensional vector, for example, where we have the basis functions defined over the anatomical regions. These anatomical regions correspond to anatomical regions within the heart. And the conductivity function is therefore approximated by the one-dimensional radial basis function approximation given like this.
You assume the conductivity is isotropic?
No. Well, yes. The thing is, it is isotropic. Given like this, it's not in the tensor. The tensor is taken as given. So the conductivity is a multiplicative factor of the tensor, and the tensor comes from the fiber, the muscle fibers.
So you assume it's known?
Muscle fibers are assumed to be known for now, yes. This morning we've seen that they cannot be found actually, which is pretty bad news. So the second question was where do we find the onset location, and the parameterization is simple. We know that the onset location will be somewhere on the endocardial surface, so it's a two-dimensional surface patch which can be done easily, and it's two parameters. So I hope I gave, so that was the partial differential equation we want to solve for, and these are the parameters that we will estimate with the cardiac data. Now the problem is what type of data we have and what are the characteristics of clinical data that we can use for the biophysical models. So the data specific for this application is the dynamic MR, which actually, as you can see here, is a 4D sequence of the cardiac muscle, taking the MR machine. We can extract, for example, the healthy muscle. We can extract the left ventricle, as we've seen in Nico's talk. We can do a lot of things. We can extract the scar regions and have a mesh basically for our computational models. What will derive the computational model, the electricity model, is basically catheter mappings. Now, these are very interesting observations. What happens is, during a heartbeat, the doctor goes in with a catheter, probes different locations within the endocardium, and observes potentials. Furthermore, he does the same thing outside the heart and observes other potentials. These points, what we see in here, are those catheter mapping points and where they touch the cardiac surface. So basically what we have here, we have, for example, the potential mappings at these points. So we've seen that the partial differential equation holds for the whole domain, and we have the electrical activity going on through the muscle. But however, we have only like a sparse number of points on the boundary. Furthermore, as I've said, this procedure is there's a catheter going within the heart as the heart is beating. So you can imagine the uncertainty going on here because the catheter is touching and the heart is moving, and this location does not really correspond to where it touches. So if we're talking about the parameter estimation problem, or these are the typical characteristics that you would have for biophysical models, the inverse problems or the parameterization methodologies should take these into account. So in order to take these into account, I will define a probabilistic framework for the parameter estimation. So rather than trying to assume point estimates for the parameters, I will try to regard the parameters as random variables. And the partial differential equation will act as a conditional dependency tree for these parameters and the solution. What I will have is basically a joint distribution of the observations and the parameters. Assume I've shown you these catheter mapping points. Assume that my observations there are depolarization times given by t tilde. Now, the joint distribution can then be written as this. So what we have here is we have the prior distributions for the parameters, basically saying that if we do a population statistics or have some kind of information, we say that these parameters are the possible ranges of what we can observe. Then we have the observation model, which basically says that if I'm at a location, if the actual depolarization time or actual potentials are this, our noise model, our observation model will distort it in some sense. And then the physiological model, or the PD, will come into play right here, where we have this for a certain set of parameters, what are the possible outcomes of the model itself. So using similar tricks, assuming independence between the observation points, we can go even further, simplify this equation, and as a result, assuming that once the set of parameters and the boundary conditions are given, the Eikonal-Diffusion models will have a unique solution. We can write down the posterior distribution simply like this. So now, let me try to give you an intuition what this posterior distribution will look like in terms of the parameters. I've created a very simple toy example here where this is my left ventricle, which looks like a cup. And I'm starting an electrical wave right here in this green region. Furthermore, I will take observations from the other side of the cardiac tissue around these red points. So my simulation is very simple, it's two-dimensional. I will have a high conductivity speed on the endocardium, so within this cup, and a lower on the muscle conductivity in the myocardium tissue. So when I simulate it, what I will have is an evolution like this. Now, this image corresponds basically to an axial slice of this cup. I simulate it here, and I observe the outputs right there. Now, what happens is, let's look at the posterior distribution. This is, once I observe the values, the posterior distribution for the parameters, right? Now, the interesting thing is the link between the observation points and the posterior distribution for the parameters. What I do is I slowly increase the number of observation points and recalculate the posterior distribution. And what we see is here there is a very close link. If I observe n number of observation points right here, I see that there is a huge dispersion and my confidence is low. And furthermore, when I increase the number of observation points, I can see that I actually narrow down in the posterior distribution. Now, the deterministic algorithms are wonderful and they can find point estimates right here, but they lack this information about how confident I am, and furthermore, what is the relationship between the different conductivities and what are the sensitivities. This was nice, this was all 2D and it was computable in a reasonable amount of time. Now, the problem we have here is what if we're talking about a higher dimensional problem? Higher dimensional as in terms of what if we have 20 parameters, 30 parameters, how those things work. Now, the problem is we have to solve this posterior distribution. So we have to know the solution for the forward problem. So this posterior distribution is analytically computable if basically we know the analytical solution for our model. But this doesn't really happen a lot. So we use sampling-based methods, or what we can do is we can use collocation-based methods, try to reconstruct the posterior distribution through sparse grid reconstructions. Well, the problem is in all these things, there's a computational bottleneck, and which is that we have to solve this model equation many, many times. Now, a very simple computation, if we had a 20-dimensional problem, we would have, for example, 10 to the 5 simulations, 20 seconds per simulation. That already makes more than 20 days. So one solution in this line is the reduced basis methods or the spectral methods. And I will detail a bit about that and show its problem and then show how we solve it. Now, reduced basis method, spectral methods are based on the spectral representations for the random variables. So what we have is we have the polynomial Keros expansions for any type of random variable, which basically means that if I have my random variable, in this case, for example, d vector, I project this onto another type of random variable, shown as psi here. And this projection is a Hilbert space, and it basically, through the theorem, we can have the polynomial expansions and have a representation theorem. The nice thing is, as a normal Hilbert space, in the respective probability space, we will have orthogonality between the polynomial bases. Furthermore, we will be able to create, again, a spectral basis for that specific random variable based on the simple projection equations. So what is the exact relationship between this and the PDEs? Well, the thing is the randomness of the PDE is caused by the randomness of the parameters. And the randomness of the solutions of this model can be represented in the same basis as the randomness of the model parameters. So what we have in the end is we can represent the random solution, the random depolarization times that comes from the randomness of parameters with the same basis, with the same expansion. And if we know the spectral basis functions, then what we can do is, for a given specific set of parameters, instead of solving the PDE numerically, now we can approximate the solutions by simply a linear combination of functions, which is, as you can imagine, much faster than just solving simulations many times. Now the problem is, relating back to our demonstrative problem, is for the ED model, for example, I can assume uninformative prior vector and have basically a uniform distributions. This uniform distribution will link to our psi variable as uniform distributions as well, and we will have very simple spectral basis, for example, linear case. And in this case, the randomness of the psi vector dictates us which polynomial basis to use, which will be the Lejeune type. So what happens in the depolarization times, the same thing applies, and as I said, we have the decomposition equations, and furthermore, the spectral basis, the TIs, actually satisfies the projection equation. Now, the big problem here is naturally how do we compute the spectral basis, right? I said if we know the spectral basis, then we can approximate the solutions of the model easily. Now, there are different methods. We can use Galerkin projections, the simple FEM-based method. But that will be harder, especially if we have nonlinearities, and if we're talking about high number of dimensions, then we can easily end up with tens of thousands of coupled nonlinear equations. We can go a numerical way and try to solve for this integral, which will basically can be done with stochastic collocation, anything. But again, we'll have the curse of dimensionality. We solve for the nonlinearity, but if we have high number of parameters, then this will cause troubles. What we use here is basically try to use somehow the smooth assumptions of the model itself or the biophysical models that are usually there. So sparse constructions help us there. For a given point on the observations, we don't really expect all the parameters to influence the solution. So therefore, actually, we expect a sparse representation in the spectral domain. And this sparse representation gives us the ability to be able to compute these basis functions or approximate these basis functions using only a small random projections of the model solution. So what does this mean basically? This means that for a high dimensional problem, say 20-dimensional problem and a complex geometry with a high resolution grid, we can compute the spectral basis functions and the spectral representation of this PDE using only a few hundred random projections. The plots I show here are basically the approximations areas between the simulations that are computed by the PDE and the spectral approximations. The simulations, each simulation you can imagine takes about 20 seconds to 40 seconds based on the fiber structure. Each spectral representation computation takes about 30 milliseconds. So we see that only with a few number of simulations, we can construct the spectral basis and we can have very accurate approximations to the solutions of the PDEs. So let's go back to our question and just try to link all these together and see what will happen with the cardiac case. What does Bayesian inference can give us for the biophysical models. One last thing that I have to talk about before going into the results is basically the observation model. And this basically links how I define my observation noise or uncertainty in my model. The basic assumption we use here is we know that there are a lot of sources of uncertainty, the motion artifacts, the estimation of the depolarization times from the potentials, or the projections of the observation points onto the mesh that was extracted in the image acquisition. So by a simple assumption, we know the projection distances, for example, between the image and the intervention domain. So we can include these kind of information and figure out, for example, yes, we have some kind of uncertainty, but it's a spatially varying noise model on the observation. Okay. If we go to the results, so this is the type of thing that we get from Bayesian inference in the biophysical models. Using conventional deterministic methods, what I would have obtained is simply this point estimate. So yes, I had the observations, I solved the Eikonal model, I estimated for the conductivity function, and this is what I get. However, when I now look at the posterior distributions, I understand, for example, I can look at the standard deviations on these estimates. And I can see, for example, for some regions, I'm much more confident, while for other regions, the variation is so high that actually I cannot really trust the values that I optimize for. The same thing actually applies for the on-state location as well. So what we have is we have this link between the data uncertainty and the quantification of this uncertainty on the parameters. When we do model fitting with these, what we end up with, the usual conventional methods gives us this point estimate of, okay, this is the result, this is the model fit. What we have here is rather the range of all possible personalized simulations that takes into account the posterior distributions for the parameters. Now, asking another question, for example, let's say we do the model fitting with a region, and now we try to predict what will happen in the other part. So what we do here is, as I said, we had the endocardial and the epicardial observation points. Let's assume we do the fitting only on the epicardial and try to predict what will happen endocardial surface. The thing is now it's pretty interesting because we also have the link between the uncertainty on the parameters, which as we see right now, the confidence levels are even lower. The link between uncertainty of the parameters and the model solutions are given. This basically shows that yes, where we do the model fitting, we might have a nice point estimate and we might interpolate between the observation points. But when we do the prediction, for example, what we notice is the ranges for the possible solutions are much higher due to these parameter uncertainties and sensitivities. If we look at another region, for example, say that we do exactly the same thing, but now do it in the endocardial surface and try to predict what happens in the epicardial surface, the results are even weirder. Now we know that, for example, the uncertainty in the parameters are lower, so we have higher confidence. But then we notice that the endocardial surface, for example, doesn't really give us a lot of information for different regions in the heart. So when we try to predict what will happen in the epicardial surface, we see that the range of the possible simulations are just way too big, so we cannot conclude anything from there. So, yep, this was basically it. And what are the challenges? So, as I said, this is kind of a nice probabilistic framework for the biophysical models to fuse different sources of information. What I've shown was basically a method to take into account the high dimensionality of the problem. But the assumption was the underlying model is smooth, or rather well behaved. But then the question is, what happens when we have multi-phase models? What happens when we have bifurcations in the model? How can we take into account? And these are open questions. This also actually gives us an interesting framework to do multi-scale modeling and somehow inference of the microscopic parameters. Usually, we know that most of the diseases and most of the structural disorders arise from the microscopic behavior of either cellular behaviors or genetic behaviors. The question is, we're not really observing these when a patient comes in. We're observing rather large-scale behavior, the images, the microarrays, or whatever you want to think of. The question is, can we use such a framework, for example, to take those observations and try to infer what might happen in the microscopic scale, the parameters which we don't observe really. Yeah, that's basic.
How do you determine the scar tissue location?
I'm sorry?
How do you determine where the scar?
Oh, that's done by segmentation, same automatic segmentation.
So you
It's
have
very interesting work. I have two questions. The first is, how do you validate actually the model? How do you measure whether or not this model fits to reality? And the second question is, what is the clinical value of having this model? Why it's useful? I understand it's a nice theoretical problem, but
Well, basically the use is right now, as I said, as I've shown above, is to be able to do simulations for the radio frequency ablation. That's the kind of the model example that we showed. But rather than the ED model, now the CMAX is actually looking for the blood flow models. And just because if you look at the blood flow models,
The first one is how do you measure the success?
His first answer, did you get that?
Yeah, I mean, it's,
so, but he wasn't very confident. So this is the radio frequency application that we show here. As I said, the other applications actually expand to fluid dynamics and try to figure out how structural deformations occur based on the pressure that's caused by the blood flow.
because, you know, there's, for example, Charles Peskin has developed this amazing model back in 90s.
Yes.
And I met him several times. Not always he's complaining because
Yeah.
he has this fantastic model based on fluid dynamics, which actually can produce anything, can produce pressure, fluid, whatever you want. But nobody's using it.
Just because it's slower.
I'm not sure because it's slower, it's because I think it's a very complex, we're trying to solve a very complex problem, which actually, we are not able to solve simpler problems
Yes.
in hard.
Yes.
And you're looking for really nice, fancy problems mathematical, which,
But this is not, I mean, what I'm showing here is not, what I'm showing here is basically a tool that can tell you if the model is good or not. I'm not talking about how to create the model here. I'm rather talking about how to assess the quality of the model. And then one of the main results that the people are excited about is actually this graph, which actually tells that the model is bad. Which basically says that I can fit the model in the endocardium perfectly, and I have a wonderful fit, and I interpolate between the data. But it doesn't tell anything about what happens in the endocardium. So if that's your question.
yeah. Still, I have tried to find out the clinical value. But I guess it will come in 5 years from now.
Maybe. Well, I guess Community's group are working on this.
Okay.
Let me ask one question. How do you determine the prior?
Right now it's uninformative, but we'll
Is there such a thing, uninformative prior?
basically a very large prior distribution taken from the minimum and the maximum values that were reported in literature. And we try to keep it as big as possible, so we actually figure out what are the effects. But right now, we're trying to do a little bit of population statistics to figure out where the prior information lies.
Is it reasonable to say that if your prior is not reasonable, it's better not use Bayesian methods, but use some frequentist approaches? Or?
Probably yes. I mean, in this case, when you use the uninformative prior, in any case, that will be some kind of corresponds to the maximum likelihood estimate or like maximum likelihood plus rate.
We can discuss this later.
Yeah.
Any more
I'm just
questions?
curious. From the inside, they use this catheter, but then you say they go on the outside of the cervix. How do they get that?
They actually make the patient wear a vest on the heart. So during the procedure, they put a vest around the heart and then they go in with a catheter. So it's a very, very invasive procedure.
Yeah, to cut them open with the vest on.
Yes, yes, no, it is an open heart surgery.
Oh, okay.
Thank you very much again.